Hello, everybody. My name is Reka Gal, and I'm a senior at Paul Laurence Dunbar High School in the Math Science Technology Center. I'm here today to discuss my capstone research project, identifying novel mechanisms of drug resistance in ovarian cancer. I conducted my research at the University of Kentucky Markey Cancer Center under the mentorship of Dr. Jill Kalesser. This is a brief overview of what I will cover in this presentation. I will start with an introduction to my research, move on to my goals and hypothesis, and discuss my methodology. I will review my results, conclusions, and future directions of my project. I will then explain applications of my research. I will cover my acknowledgements, references, and open the floor to any questions. Drug resistance is a process in which cells that have been exposed to a substance develop changes which alter the cell and its response to said drug. As cancer cells are exposed to drug treatments, in some cases, drug resistance to those targeted therapies develops. This results in the cell becoming less effective at producing its intended outcome in the cells, namely killing the cancer cells. In the clinical setting, it is prognostically negative to see a patient establish drug resistance. In this experiment, I attempted to induce drug resistance to determine its mechanism. The IC50 value is the half maximal inhibitory concentration, which is a measure of the potency of a substance. In other words, the IC50 is a concentration at which 50% of cells die due to treatment and is a measurement for resistance. In spite of recent advances in cancer research, ovarian cancer, which will be referred to as OC in this presentation, is one of the top 10 leading new cancers for women in the United States and is also one of the top 5 causes for cancer-related deaths. In 2018, ovarian cancer was estimated to account for 14,070 deaths in the United States. One of the primary reasons for the high mortality rate is that ovarian cancer is often not diagnosed until an advanced stage. Moreover, a high percentage of patients will relapse after first-line chemotherapies. Because of this, developing targeted therapies for ovarian cancer has become a priority. Approximately 30% of ovarian cancers have mutations in the BRCA1 or BRCA2 genes, key players in DNA double-strand break repair. When BRCA and other homologous recombination components are mutated, cells will become dependent on other mechanisms of DNA repair. PARP inhibitors, such as olaparib, block one of the key components of base excision repair. A normal cell will switch to using homologous recombination for DNA repair after treatment with a PARP inhibitor. However, if the homologous recombination pathway is already compromised, then blocking base excision repair will result in cell death. PARP inhibitors have shown great success in homologous recombination mutated ovarian cancer, but many patients will ultimately relapse. The BRCA1 null OC cell line, UWB1289, was utilized to establish olaparib-resistant cell lines. Epithelial cancers have been linked with increased mortality rates, recurrence of cancer, and a loss of drug efficacy during treatment. The majority of patients with advanced stage OC will develop resistance to first-line chemotherapies and targeted therapy, highlighting the importance of identifying new therapies for OC. My investigative question was, what mutations in OC are associated with resistance to PARP inhibitors? And my hypothesis was that next-generation sequencing will reveal novel genetic alterations in olaparib-resistant OC cell lines that will predict sensitivity to targeted therapy. I sought to address this problem by developing clinically relevant olaparib-resistant ovarian cancer cell lines in order to identify novel targeted therapies. The resistant line was generated by treating the UWB1289 cell line with either a continuous or pulsed clinically relevant dose of drug. For the pulse method, cells were treated with a specific dose for the 48 hours. Drug treatment was repeated every 14 days with approximately 3 to 5 cycles, or until cell lines showed signs of resistance to treatment, such as an increase in proliferation or change in cellular morphology. Cell lines were then tested for drug resistance by comparing the IC50, drug concentration for 50% growth inhibition, of the predicted parental line to that of the parental cell line. Predicted drug-resistant cell lines and the parental cell line were tested for cell viability using a luminescence cell viability assay. All experiments were performed in triplicate. Cell viability numbers were normalized, and the IC50 value for each cell line was calculated using R software. The IC50 values of potential drug-resistant lines were compared to that of the parental cell line. A twofold or greater change in IC50 was considered significant. The y-axis displays percent cell viability. The x-axis displays concentration of olaparib in micromolars. The graph indicates drug concentration required for 50% growth inhibition of the parental cell line, which is shown with the green line. The UWB1R1 cell line was developed by treating UWB1289 cells with 0.5 micromolar olaparib for 3 weeks. The UWB1R2 line was developed by treating cells with 1 micromolar olaparib for 8 weeks. Cell variability assays were performed on the parental line and the putative resistant lines. UWB1R1 showed a 2.7-fold increase in IC50, which is shown in the red compared to the parental line, which is shown in black, while UWB1R2 showed more than a 6-fold increase in IC50, which is shown in blue compared to the parental line. Next-generation RNA sequencing was performed on the R1 resistant line and the parental line. I utilized QIAGEN's Ingenuity pathway analysis to analyze the RNA sequence results. That suggested the retinoic acid pathway was altered in the resistant line at retinol binding protein 1 and retinol binding protein 7. Based on this, I decided to test if these cell lines were sensitive to the UAB30 RXR agonist. UAB30 is an anti-cancer differentiating agent. I treated the parental and the R1 resistant cell lines with different concentrations of UAB30, then performed cell proliferation assays at 96 hours. The y-axis displays percent cell viability, and the x-axis displays the concentration of UAB30 in micromolars. The graph indicates drug concentration required for 50% growth inhibition of the parental cell line. The UWB1R1 cell line was slightly more sensitive to UAB30 than the parental line. The dose-response curves shown here are the average of triplicate experiments. By 96 hours, all the UWB1 cell lines showed sensitivity to UAB30. The UWB1R1 and UWB1R2 cell lines exhibited increased resistance to PARP inhibition after constant exposure to the drug, displaying over a 2-fold increase in the IC50 compared to the parental line. The UWB1R2 line, which was developed at a higher drug concentration over a longer exposure time, demonstrated increased resistance to olaparib when compared to the UWB1R1 line. The data presented in this study provided partial support for the hypothesis that next-generation sequencing would reveal novel genetic alterations in olaparib-resistant OC cell lines that would predict sensitivity to targeted therapy. The UWB1R1 line was more sensitive to the RXR agonist UAB30 than the parental line, which implied that there was an association with resistance to PARP inhibitors. Further sensitivity testing may provide important information on the development of resistance. These results indicate the need to pursue novel synergistic drug combinations. This would allow for multiple pathways to be targeted to achieve maximum efficiency in reducing resistance in the UWB1 cell line. Potential combinations include UAB30 with anti-apoptotic inhibitors, such as BCL2 inhibitors and SRC kinase inhibitors. The results from the RNA sequencing could also be further analyzed to identify novel putative drivers of olaparib resistance. Novel synergistic drug combinations will be tested in mouse models of ovarian cancer. These results are applicable to the future of precision medicine. By sequencing a patient's genome before treatment, specific pathways may be targeted by providing the individual with the most effective drug and decreasing resistance based on their mutations. Furthermore, establishing the causes of drug resistance in OC cell lines at various stages of resistance with next-generation sequencing may provide important insights into how to modify the treatment based on the progression of an individual's cancer to reduce or prevent resistance. I would like to thank everybody who has made my research possible. I would like to thank Dr. Joe Kolesser for mentoring my work and members of the Kolesser Lab, especially Dr. Abigail Anderson and Dr. Brian Burgess for their guidance. I would like to thank Ms. Karen Young and the Math, Science, and Technology Center for guidance and encouragement. I'm grateful to the Kentucky Young Researchers Program for the funding portions of my study. I would like to thank Dr. Clinton Grubbs for providing UAB30 for the study. And last but not least, I would like to thank my friends and family for their support in Countless Carbide's lab, specifically Taylor Albright, Kelsey Siebenthaler, and Praneeth Bodden. These are my references. All images that were not cited were self-generated. If you have any questions about my research, please feel free to contact me at the email shown on screen. Thank you very much.